Thanks for coming. So my name is Brian He. I'm very excited to share some of the work we've been doing on developing large language models or sequence models of molecules, but also potentially how these molecules can interact with each other and also interact with the broader genomic context within the biological organism that these biological molecules exist in. So to begin, all of you are probably aware that machine learning has and is continuing to revolutionize the study of molecules. So for example, AlphaFold is a program for predicting the structure of protein molecules either as a monomer or proteins bound to other proteins, or maybe proteins bound to molecules like nucleic acids like RNA or DNA. So we're still very much interested in molecules, but we're also interested in how these molecules basically come together to form a very complex biological organism like a whole cell or even a single-celled organism. And when you actually think of things at this level of complexity, the complexity required to understand things at the whole cell or whole organism level is actually several orders of magnitude more complicated than any individual molecule. So this is very difficult. How would you even begin to tackle this problem? How do you even get enough training data to reason about things at this level of complexity? So it turns out that the DNA sequence of an organism does contain the information for all of the protein and DNA and RNA molecules that come together to make an entire organism. And actually over the past 20, 30 years, people have been sequencing the whole genomes of various organisms and just depositing them publicly available online. And so there's a lot of whole genome sequencing data that's publicly available, and all of this information does encode information about the molecules that come together to create a whole organism. The one potential problem, though, is that when you look at the DNA sequences in these databases, for example, it basically looks like a string of A's, C's, G's, and T's that if you had no kind of domain-specific knowledge, or even me, if I just look at the genome, and I've been doing this for a while, it just looks to me like a random string of A, C, G, T. There's, I don't speak DNA, for example. But actually there's a lot of patterns, there's a lot of structure in these genomes that we can hopefully untangle using modern statistical machine learning methods. So kind of borrowing from recent progress in machine learning for natural language, in which you train models that will predict the next word in a sentence, or the next token in a sentence, given some sentence prefix. We're also going to apply this kind of pre-training strategy to predict the next base pair in a DNA sequence. And actually, in order to predict the next token well in natural language, the model will have to understand things about the grammar of a sentence, or maybe even higher order semantic concepts of a sentence. And analogously, if you want to predict the next base pair well, the model will have to understand some of the statistical rules that go into how evolution puts a nucleic acid base pairs together to form a DNA sequence. So we have the data, we have all these whole genome sequences that have been publicly deposited, and we have a pre-training task. So we're going to predict the next token in a sequence of base pairs. So what kind of model do we use to do this? So this is a model that we've trained that we're calling EVO, and it's a long context genomic foundation model. So basically what this model is doing is it's predicting the next base pair given a sequence of bases. But there's a couple things that are very special about this model. So this model is trained with a new architecture. It's called striped hyena. And basically what this architecture lets us do is process a very long sequence of base pairs in the model's input. So we're actually able to process 131,000 base pairs in the input. And it's also, the model itself is actually quite large in terms of the number of parameters. So we train this model for 10 to the 22 floating point operations. It's 7 billion parameters, and it's trained on 300 billion tokens. Each token corresponds to a single nucleotide. So it's single nucleotide resolution. The important thing about this architecture is that it enables efficient scaling to these long sequence lengths, but also it enables better understanding of things at a single nucleotide resolution, potentially like in the kind of inductive bias of the model, being able to group different tokens together and kind of reason about things without us having to explicitly enforce some kind of higher order tokenization. Cool. So this is a track on molecules. I just wanted to emphasize the different scales of complexity that the model can actually understand. So if you think about models of molecules or common biological molecules like proteins or RNA, so for example, Cas9 or lysozyme, which are these proteins, or a tRNA, which is a very small RNA, they're actually quite short, so you can maybe understand them using programs like AlphaFold or ESM, which have these pretty short context lengths at the molecular scale. But a problem is that if you want to understand information at, say, the scale of whole genomes, or even these gene clusters that have in which there are multiple molecules working together to accomplish the same function, then these small genomes or these gene clusters are actually much longer contexts than anything you can fit into the context window of ESM or AlphaFold or something. But if your model has 131,000 base pair contexts, then in theory you could also process all the information needed for these different genomes in the model itself. So like I said before, we train the model on these bacterial and phage genomes that have been publicly deposited. It corresponds to a total amount of 300 billion nucleotides. We have around 80,000 whole bacterial genomes, and then around 2.7 million other genomes from viruses or plasmids. But we also do exclude eukaryotic viruses in the training data for this particular model, mainly for safety-related considerations, actually. Another thing that I wanted to highlight is that in natural language, it's been shown that if you just increase the number of parameters in the model, and assuming you have a very large data set as well, increasing the number of parameters in the model actually somewhat magically corresponds to a better ability to predict the next token. So this is sometimes referred to as scaling laws in natural language. So actually for this study, we also wanted to see if training on DNA sequence would benefit from the same kinds of scaling laws. And so the takeaway from this slide, I know there's a lot of data on here, but the takeaway from this slide is actually that the same scaling laws do apply to DNA sequence as well. So actually, if given the complexity of the data set that we use to train the model, if you just increase the size of the model and the number of parameters, and increase the amount of compute that you use to train the model, this will translate into very predictable ways to predict predictable performance improvements in predicting the next token with the scale of the model. So it's good to see. And it enabled us to basically scale the models to 7 billion parameters without us, being worried that we would waste a bunch of compute, for example. So one reason why I'm very excited about training models on DNA sequence is that the DNA sequence in the genome of an organism, because of basically the beauty of the central dogma of molecular biology, the DNA sequence actually encodes all of the DNA, RNA, and protein molecules that come together to form a whole organism. So actually, if you just train, say, a long context genome foundation model, then in theory, it can also learn information about these different modalities across the central dogma of molecular biology as well. So we actually do show this. So there are these different studies where people will actually make mutations to a protein and then experimentally measure the effects of these mutations on some notion of the protein's fitness or how well it accomplishes some specific function. There's analogous studies like this for RNA and DNA as well. So people do have shown that if you train a protein language model, for example, and you just take the likelihoods that a protein language model assigns to a given protein sequence, when you make a mutation, and you do this for many different mutations, the language model likelihood actually correlates pretty well with experimentally measured fitness of these proteins. So this is one way people in these different domains actually benchmark their the quality of their different molecular language models, basically in the ability to predict in a zero-shot way, just directly from the pre-trained model likelihoods, the correlation with fitness, or the predictive performance in predicting fitness. So actually, very interestingly, even though we train the model on just long genome crops, so we don't give the model any explicit annotations of where the protein coding sequences are, or we don't tell it where the non-coding RNA sequences are, or where any of the regulatory DNA sequences are. The model can actually learn some properties of protein function directly from the genome sequence. And this ability, for example, to predict the mutational effects on protein fitness is better than other models trained on genomes directly on DNA, and it's actually approaching. I guess this didn't project very well, but it looks great on the computer. But well, you can maybe see the black dots. It's approaching in terms of the predictive performance. It's competitive with models just directly trained on proteins. But I guess this didn't project very well, so sorry about that. Something that we're also excited about is, let me just try to, I can highlight. Okay, so there are these blue squares here that correspond to different coding sequences. This also didn't project super well. So something we're interested, or that we saw that was pretty interesting to us is actually we can also predict the effects of mutations on proteins, but in a larger genomic context. So let me explain this experiment that we did. So we basically take either a very long genomic sequence. So it's the entire genome of a phage, for example, or a very long genomic crop of a bacteria. And then we're going to make at each gene in the genome a single stop codon insertion. So just 3 base pair premature stop codon insertion. This is a deleterious gene if you actually made this, deleterious mutation if you actually made these mutations in the organism, because you just put a stop codon at the beginning of the protein. So we're going to do this for each gene in the genome. And then we're going to compute the EVO language model likelihood of the mutation and compare it to the mutated sequence, and compare the likelihood to the likelihood of the unmutated sequence. And when we do this, we tend to see that if you put one of these deleterious mutations in the sequence of an essential gene, the language model likelihood drops a lot more than if you put the mutation into the sequence of a non-essential gene, indicating that the model somehow knows that it's okay to maybe mutate or kind of knock out these essential or non-essential genes, but it's less good to do this for the sequences of essential genes. And so this is showing us that the model can actually kind of reason about the function of these different protein coding sequences in a broader genomic context, which is pretty cool. But one thing that we're especially excited to do with these models is that because they're trained autoaggressively, that we can also use them as generative models to, say, generate a new DNA sequence. And something that we wanted to highlight is these models can also design DNA sequences that have multiple genes in them, and each of those genes can also encode molecules from different molecular modalities too. So for example, the CRISPR-Cas system for doing genome editing requires a very large protein molecule that's around 1,400 amino acids long, also bound to a guide RNA. So it's a very large protein bound to an RNA, and that complex itself is required to actually do the genome editing. So we actually use the model and fine-tune it on these CRISPR-Cas sequences, which, fortunately, in the genomes of bacteria, the CRISPR-Cas protein is often directly next to the RNA that it uses to edit the genome. So just getting this to work was actually quite challenging to fine-tune the model to generate such a long protein, but we can at least generate sequences with predicted structures from AlphaFold that adopt similar structures to Cas9, as well as we can generate the guide RNAs to have the right kind of secondary structure as well. And just to emphasize that these proteins are actually quite large, so getting coherent generation at this sequence length was already quite tricky. And the sequence lengths of Cas9s actually exceed the context lengths of a lot of protein language models that are 1,024 amino acids in their context. Here we can do generation, in this case, up to 8,000 base pairs. Another reason why we're excited about this is right now, if you want to basically generate diversity in genome editing systems like CRISPR-Cas, what people do is they'll just mine genomes from nature. So they'll sequence the dirt, or they'll sequence deep sea vents and look for new kind of CRISPR-Cas systems. But another reason why we're excited about this approach is we could potentially replace some of this genome mining with a generative model instead. A similar thing we did for these systems called mobile genetic elements. So these are actually very minimal self-replicating systems. So for example, this is the IS605 mobile genetic element that encodes 3 different genes, so 2 proteins. And these proteins don't actually bind to each other in order to accomplish the function of the MG, but they do work together. And one of these proteins also binds to this omega RNA. And we can also show that we can get coherent generation of these systems in terms of their AlphaFold predicted structures or their predicted RNA secondary structures, where we have all the right components in place. Another kind of more crazy thing we did is just try to generate unconditionally from the model, or generate from the model at much longer lengths than even its context, its sequence context. So the model has 131,000 base pairs in its context, and we use it to sample sequences that are 650,000 base pairs long. When we did this, a surprising thing we found is that the generations from the model actually, at a very high level, kind of sort of resemble prokaryotic genomes in nature. So for example, they have the right coding density. So when you go and you predict the open reading frames, or the protein coding, putative protein coding sequences in the genome, actually the genomes generated by this model are very coding rich. And another thing that we observed is in natural genomes, a lot of genes will actually cluster together on the same strand. And this corresponds to things in bacterial genomes sometimes called operons, in which genes are kind of co-regulated with each other on the same strand. And we actually kind of see operon-like structure too in the evo-generated genomes, which we don't see in, say, just a random sequence of nucleotides. And when we also run the protein coding sequences, the putative protein coding sequences through a structure predictor, they at least show some evidence of secondary structure, lots of alpha helical secondary structure, actually. So we're excited about this general research direction as kind of a vision for the lab and this research program moving forward. And we're really excited about developing this foundation model for biology. And we use this term somewhat deliberately in that, for example, if you spend a lot of work developing a protein language model, which is something that I've worked on in the past, even if you get a really good protein language model, that might not necessarily transfer at all to, say, RNA, for example. But if we do a really, really good job at developing a genomic foundation model, then we can potentially, hopefully, impact tasks across these 3 different areas of the central dogma, so DNA, RNA, and proteins, as well as if we do a really good job at developing a genomic foundation model, we can also help out tasks from the molecular to the systems to the genome scale as well. So potentially lots of impact from this research direction. We do have a preprint available, and we've made the model weights and all the code for doing inference open source. So I just wanted to direct you to the link to the GitHub and the link to the paper here as well. And finally, this work was done with a very, very talented group of co-authors and collaborators from a lot of different institutions. So I just wanted to acknowledge them here. And thank you all for your attention. So.